They have to have a wiring diagram of all these neurons, not only in the brain, but in the body. Neurons are body-wide. The brain connectome is specific to the brain. These maps are available to international databases in various levels of detail.
We're going to Google brain connectome. And we're going to click on the Wikipedia page. A connectome is a comprehensive map of neural connections in the brain and may be thought of as its wiring diagram. These maps are available in varying levels of detail. A functional connectome shows connections between various brain regions, but not individual neurons. These are available for large animals, including mice and humans, and are normally obtained by techniques such as MRI, and have a scale of millimeters. At the other extreme are neural connectomes, which show individual neurons and their interconnections. These are usually obtained by electron microscopy and have a scale of nanometers. They are only available for small creatures, such as the worm and the fruit fly, and small regions of mammal brains. Finally, there are chemical connectomes, showing which neurons emit and are sensitive to a wide variety of neuromodulators. As of 2025, only the worm has such a connectome. Now let's scroll down. Throughout the 2000s, several investigators have attempted to map the large-scale structural architecture of the human cerebral cortex. One attempt exploited cross-correlations in cortical thickness or volume across individuals. Such gray matter thickness correlations have been postulated as indicators for the presence of structural connections. A drawback of the approach is that it provides highly indirect information about cortical connection patterns and requires data from large numbers of individuals to derive a single connection dataset across a subject group. Other investigators have attempted to build whole brain connection matrices from DW MRI imaging data. The Blue Brain Project attempted to reconstruct the entire mouse connectome using a diamond knife sharpened to an anatomic edge and electron microscopy for imaging tissue slices. They ended up in 2018 with an atlas providing information about major cell types, numbers, and positions in 737 regions of the brain. Now let's go over here, and we're going to read Genetic Influences on Hub Connectivity of the Human Connectome. Brain network hubs are both highly connected and highly interconnected, forming a critical communication backbone for coherent neural dynamics. The mechanisms driving this organization are poorly understood. Using diffusion-weighted magnetic resonance imaging in twins, we identified a major role for genes, showing that they preferentially influence connectivity strength between network hubs of the human connectome. Using transcriptomic atlas data, we show that connected hubs demonstrate tight coupling of transcriptional activity related to metabolic and cytoarchitectonic similarity. Finally, comparing over 13 generative models of network growth, we show that purely stochastic processes cannot explain the precise wiring patterns of hubs, and that model performance can be improved by incorporating genetic constraints. Our findings indicate that genes play a strong and preferential role in shaping the functionally valuable, metabolically costly connections between connectome hubs. So, what is a brain network? We're going to go over here to Science Direct, and we're going to read network hubs in the human brain. Brain hubs can be detected with tools and methods of graph theory. Network studies have consistently identified structural hubs in cerebral cortex. Hubs are central in brain communication and neural integration. Hubs participate widely across a diverse set of cognitive functions. High centrality makes hubs susceptible to disconnection and dysfunction. Virtually all domains of cognitive function require the integration of distributed neural activity. Network analysis of human brain connectivity has consistently identified sets of regions that are critically important for enabling efficient neural signaling and communication. The central embedding of these candidate brain hubs and anatomical networks support their diverse functional roles across a broad range of cognitive tasks and widespread dynamic coupling within and across functional networks. The high-level centrality of brain hubs also renders them points of vulnerability that are susceptible to disconnection and dysfunction in brain disorders. Combining data from numerous empirical and computational studies, network approaches strongly suggest that brain hubs play important roles in information integration, underpinning numerous aspects of complex cognitive function. Structural hubs. Compiling macro-scale connectome maps of the human brain from diffusion imaging data. Not only can they see what you're thinking, they can see how your brain is putting it together with the wiring map. Let's go over here and read molecular communication, harnessing biochemical materials to engineer biomimetic communication systems. Molecular communication uses molecules, like biochemical signals, as an information medium and allows biologically and artificially created nano or microscale entities to communicate over a short distance. It is a new communication paradigm. It is different from the traditional communication paradigm, which uses electromagnetic waves, like electronic or optical signals, as an information medium. Key research challenges in molecular communication include design of system components, like a sender, a molecular propagation system, a receiver, and a molecular communication interface, and mathematical modeling of each system component, as well as entire systems. We review all research activities in molecular communication to date, from its origin to recent experimental studies and theoretical approaches for each system component. As a model molecular communication system, we describe an integrated system that combines a molecular communication interface using a lipid vesicle embedded with channel-forming proteins, a molecular propagation system using microtubule motility on kinesin molecular motors and DNA hybridization, and a sender/receiver using giant lipid vesicles embedded with geminine peptide lipids. We also present potential applications and the future outlook of molecular communications. Communication in biological systems is typically achieved by molecules, except for electrical signal transduction mechanisms, such as nerve systems, and is often found in aqueous environments, such as within or between biological cells. For instance, multicellular organisms, including human beings, perform maintenance of homeostasis, growth regulation, kinematic control, memory, and learning through inter and intra-cellular communication using signal transducing molecules such as hormones. If artificially designed and controllable molecule-based communication systems that transmit biochemical information, like the status of living organisms, can be developed, which is not feasible using traditional communication, it may be possible to devise a new communication paradigm. In 2005, we named this concept molecular communications and have pioneered the research in this field. Molecular communication provides molecule-based biomimetic communication mechanisms, and it allows biologically and artificially created nano and microscale entities, like biological cells and biohybrid devices, to communicate over a short distance. Molecular communication has received increasing attention as an interdisciplinary research area, which spans nanotechnology, biotechnology, and communication engineering.